Okay, welcome to this lecture here on introductory genetics. So, first I'm going to ask the question, what is a gene and how do genomes differ between prokaryotes and eukaryotes? So first off, what is a gene? Well, a gene is a basic unit of hereditary information that codes for RNA. So moving on with what we know about this, what are the differences between prokaryote genomes and eukaryote genomes? Well, prokaryote genomes are smaller. There are no activators, so there is no increase or decrease in expression as much. There are no introns, exons. What gene is read is actually produced, and later we will find that prokaryotes actually do have introns, but they are self-splicing and do not work as well as in eukaryotes, and are not as prevalent. So now looking at eukaryotes, they have large genomes, they have many introns, exons, and they have activators that decrease and increase expression. So moving on here to prokaryotes, we're just going to look at them in depth for a little bit. So prokaryotes have a promoter sequence, they have a coding region, and a terminator sequence. So let's look a little bit more in depth at So it has consensus sequences as target sites to help bind transcription factors that allow RNA polymerase to bind at a transcription start site. Now looking at the coding region, it is a short, untranslated region and translated region. The untranslated region, used as somewhere to bind, initiates translation. So the untranslated region is used as somewhere to bind for initiators for translation. And as I said before, prokaryotes do have self-splicing introns in this region. Now taking a look at the terminator sequence here. So there's actually two main methods that polymerase can be terminated from the sequence. And this includes the row-independent and row-dependent methods. So the row-independent is basically an RNA strand forms as RNA polymerase comes along the sequence here. The strand continues to form, and then it ends up hairpinning in on itself and displaces the RNA polymerase off of this the sequence here. Now row-dependent is where polymerase then attaches yet again, creates a sequence that comes off as transcribed, and this transcribed sequence binds to a row protein that then moves up the sequence, up the transcribed sequence, and actually ends up knocking off RNA polymerase so that it's then again terminated. So now I'm just going to look on here at eukaryotes more in depth. So keep in mind here that raw resources from historic genetic merging of DNA and extra energy from mitochondria allow eukaryotes to have more complex energy taxing replication systems. So the transcription start site and a transcription stop site are contained in this long sequence that eukaryotes have that code for proteins. And within this region, there are introns and exons, and these introns can be taken out using spliceosomes. So there are actually target sequences within introns and exon region, which allow cells to know where to splice. So there are small little sequences, and they actually can't be consensus, so scientists don't know exactly what these sequences are because they change very often. And so there is no like average base pairs that can be seen that act as target sequences. So it's very difficult to realize where these splicing can occur but through actually going through laboratory tests and such, they are able to find these locations by seeing what gets spliced. And also, you would want to keep in mind that these target sequences are very small, and it is very, very, very possible that there is more to the splicing mechanism than just these target sequences that are found between the exons and introns. So moving on now to the poly-A tail that gets added. The consensus sequence for the poly-A tail is the consensus sequence for the poly-A tail addition is the transcription stop site over here. And now just a little bit of information on the poly-A tail here. It is it's the transcription termination site. It helps stability when the mRNA moves around in the cell, and it also stops it from folding in on itself as easily. It's also a tag for transporting, so other signal molecules can understand what this poly-A tail is and can recognize it and move it along to different parts of the cell. And it is the translation termination site. So once it's coded into the mRNA strand, and once the tRNAs begin to bind to the ribosomes and it goes through translation, the translation ends up stopping at this poly-A site. So now moving on to here, this 5' cap we're going to look at more in depth. So it helps tag for transporting, just as the poly-A tail. So there are certain proteins and signal molecules that recognize this cap, and it tells these signal cells that this strand of mRNA is actually intact and can be used for further processing. So the translation, it's used as the translation initiation site. So yet again, when it binds to the ribosome and tRNA, it's able to initiate the translation at this point. It helps with stability. It helps unwind RNA from secondary structure that it might turn into. So this strand here may turn in on itself and create some kind of different structure other than this linear form as shown here. And it helps make sure that it doesn't end up folding in on itself. And it also has been shown to help splice the first intron. So the intron here is actually aided by this 5' cap. And just like, as I said before, that the target sequence is not the only thing that allows these introns to be spliced. There are other things, and this 5' cap is actually one of those things that help that to occur. Introns, exons, and alternative splicing. So a cell can alternatively splice introns to create different genes. So it can also change where and how exons are left in this main sequence here. And it can also shuffle these exons back and forth. So as shown here, this pink exon can be placed before the blue exon, and the blue exon can be placed behind the green exon, etc. Introns actually vary a lot, so there can be like 0 in a gene, there can be few to very many in a genome, and these introns can be very small or they can be very large in size. There is actually a correlation between cells with larger introns. They actually have larger exons, which is interesting. Scientists don't really know yet why. Presence of introns increase expression of genes. So these introns may actually... So by saying this, I mean that the introns can also act as kind of alternative promoters. In genes, it is common to have some exons consistently present. So like, this is usually a regulatory domain or a DNA binding domain, because these things are very important to keep maintained from organism to organism to make sure that these proteins that need to be created are created. So that cells can have different promoters and sex-determined changes in splicing. Can also get partial splicing, where only part of the exon is expressed, leading to subfunctionalization. And all this really means is that the gene may have had many roles, but if it is cut into one specific role, it can do the specific job better because it is not constrained by other roles. So it's kind of like if you're multitasking, it's easier to do one job and do it better if you only have one job than it is to do that job while multitasking. You won't get that job done as well. So this is kind of evolved from endosymbiosis, where each cell had genes with the same, well, may have had, and we're very likely to have had the same genes used in an endosymbiosis event. One cell has certain genetic material, and so does the other, and it's very possible that there is overlap of genes. And so, over time, when these genes come together and form one single organism, and the genetic material is transferred, the cell may either lose some of the multiple genes, or the genes could actually adapt subfunctionality and end up more efficient. So now moving on here to translated and untranslated regions. So as you can see here, this is the untranslated region, UTR, and this here is the translated region. As you can see, proteins are being created here. So that's considered the open reading frame, or ORF for short. And here again is another UTR. So the ORF, or the open reading frame, gets translated from mRNA, and the UTR do not get translated. So these guys do not get translated. As shown, nothing comes from them. And as shown here in the open reading frame, proteins have come from these genetic sequences. So why would we then have these UTRs, and why are they of some significant size? Well, on the 5' end here, this UTR can actually have introns that control translation and influence how much translation occurs. There is actually a region in here that can actually be translated. What's been actually realized with the proteins created in this region is that they don't have much significance in the cell, and they're often degraded very quickly. But what has been realized is actually that the creation of these proteins in this area actually represses the translation of the main open reading frame. So the proteins being created here are actually repressed. So it becomes a powerful control over the open reading frame. It is likely only used as control, as I said before, the proteins created in this region actually don't have much of a purpose. And now on the 3' UTR end, it can also control translation, and this UTR here is also used for tagging mRNA for transportation, and it also improves stability. So that's why it would be needed. So now moving on here, What causes difference in cells? Well, that of course is regulation of the genes within the cells, because we know that the cells are all similar in the body, give or take some mutations here and there, but they're mostly very, very, very similar. And so across entire genes and through time, early and late development, is where the regulation of genes can actually change. So how exactly are these genes regulated? Well, one main regulator is the promoter sequence. And so it's a consensus sequence that binds polymerase, and when genes diverge, the regulation regions diverge first, instead of the coding region. So if two organisms were to slowly come apart with genetic sequences, so their sequences start to get different or diverge, as I should say, the first thing to actually diverge would be these coding regions. And this just goes to show that the actual proteins and things don't really change much, but the regulation, as the promoter changes, does change. And so these proteins will or will not be increased or decreased in expression. So this simple divergence in this regulation region can even create new species. So now an interesting thing to note is that if you put a promoter and coding sequence into a different organism, it will not be transcribed, because there's more regulation factors than just the promoter. And now this includes enhancers, suppressors, and insulators. So as shown here, there's an enhancer here, and what the enhancer does is it can be found before and after the coding region. It can be even found far away from the coding region, and it can be found inside the gene. It's not directional, so no matter where the gene is being transcribed, it can still work. And there is no consensus sequences for this enhancer. And it has been found that enhancers actually work in prokaryotes as well. So all this enhancer really does is it enhances the transcription of this certain gene coming up, or the coding region, I should say. And it does this through many different avenues, but that's a topic for another discussion. So now moving on to suppressors. Suppressors suppress transcription when bound by inhibitors. And there's also insulators, which are found between the enhancer or suppressors and the promoter sequence. And what they do is they block the suppressor or enhancer from affecting the genes. They're especially needed when nearby genes have different transcription patterns. So if you think when genes are located close by, it could be an issue if they can both be expressed by similar enhancers or similar modification of the gene. And so it is important for insulators to be here to make sure that if these two genes should not be transcribed, that only one or any certain number of them can be expressed when needed. Alright, that's the end of this lecture, and thanks for watching.